What's up, guys? We're back at it again. Got a couple of videos left, and we'll have everything posted for you for our EOC. As of today, for this recording, we're 2 days away from the EOC. So we're going to jump straight into it with meiosis, share through our review, and see what we can make happen here. Y'all, as we go through meiosis, the first thing to keep in mind is that meiosis seems very similar to mitosis. We're actually going to do a T-chart down here at the bottom that's going to help us split some differences between them. But meiosis itself is very important specifically to the idea of sexual reproduction. When we do mitosis, we make a bunch more cells for our body, and we want those body cells to match our body. If we got an injury and we need to replace some skin and muscle cells, we would want those to match the ones that we already have because that's going to allow us to stay functioning as best as possible. But when we do sexual reproduction, the last thing we actually want is for our kids to be exactly the same as us. We want genetic variation because that's going to give our species a better chance at survival. So as we look at meiosis, we're going to be focusing in a lot on what gives genetic variation in hopes that this can kind of, I guess, explain why we would need a completely similar yet different process when it comes to making cells. So we start over by jumping directly into it. How does crossing over lead to more genetic diversity? Actually, let's do something else first. I can't spell, but this can. I just want a diagram here. Something to keep in mind with this. Cell and interphase, notice that all of the chromosomes look like sticks. As it goes through interphase, it'll hit the S part of interphase where it copies its chromosomes. All of this is standard. You can now see that we have X's. We go through our first division, which is going to be called meiosis 1. In meiosis 1, we're going to be splitting our homologous chromosomes away from each other. That is the mom and dad version of the same chromosome into separate cells. You can see here we've got our two cells made from that. And if I look at these original cells, this has 4 sticks or 4 chromosomes. This has 4 chromatids or 4 chromosomes. But when I look at them, I can see that instead of just having red or blue sticks, we've got some variation already. That's going to be the crossing over that we're going to be mentioning here in a moment. The next thing to do with meiosis is to skip copying. We're not going back to interphase. We're immediately going to skip copying and go straight to a second division. When we do this second division, we're going to be taking these X's and pulling those chromatids apart so that we end up with 2 chromosomes in each of our cells here. Now, if we do a little bit of accounting, we had 4 chromosomes in the original cell. We then copied everything, bringing it up to 8. We did our first division, which brought it back to 4 chromosomes in each cell, albeit in different forms of an X as opposed to just the sticks. We skip our copying, bringing this down to only 2 chromosomes per cell. And if we look back at our original cell here, original cell had 4 chromosomes. Final daughter cells have 2 chromosomes. That gives this first one twice as much as our daughter cells. We call this diploid for having 2 full sets. We call this haploid because haploid is going to be our single set of chromosomes or half of the amount that would normally be there. Haploid sounds like half. Diploid has di for 2. We need to be able to keep that straight. Our body cells exist as diploid cells, but our gametes, the sperm or egg cells we produce, exist as haploid cells. With that said, let's go back into our review and see what we can do here. Describe how crossing over can lead to more genetic diversity. Gotcha. Okay. Crossing over happens during prophase 1. You guys, the number of times I've seen that question specifically show up on the EOC, they make a huge deal out of crossing over and genetic diversity, and they're just like, oh, yeah. So I'm just going to tell you crossing over is when they trade arms and legs and we get genetic diversity. When does it happen? Common thing to come up. So keep that in mind. Prophase one for crossing over. That's the only time the homologous chromosomes will be next to each other for them to be able to trade legs. Now that we've said that, let's actually say it directly. Crossing over when the mom/dad chromosome trade arms or legs. This does not have to happen, and whether or not it does is kind of random. But and which portions will cross and how much of those will cross, all of that's random. Maybe it'll only trade just the little tips. Maybe it'll trade halfway up the leg. Maybe it'll trade all the way up to the meeting point. We don't know. It just depends on how it plays out. Now, how does this lead to more genetic diversity? Instead of getting a chromosome that XIMILAR similar to a full mom/dad chromosome, we would receive a shuffled combo chromosome of mom and dad mixed. This makes new genetic combinations that we did not have before. Technically, we had two separate pieces. Mixing two parts of it together is what's giving us the new combo. Question 2, in your own words, explain why meiosis is significant to sexual reproduction. I will definitely do that, but then I'm going to add another little thing on the side here. There's something else that gives us genetic variation, kind of like crossing over, that I don't really see in our review, so I'm going to make sure to add that here. But let's answer 2 first, and then we'll add this on as a secondary portion. In your own words, explain why meiosis is significant. I see 2 main things. The first thing that I see, it gives us genetic variation through crossing over, law of segregation. By the way, law of segregation is just the idea that each parent only gives one copy of a gene. So if we were talking about something like a genotype and it's like, oh, for that trait, I'm big B, little b. Law of segregation would say that one of these has to come from mom and one has to come from dad. No parent would give both of them. So we segregate those 2 alleles away from each other so that the other parent provides one of them. That's another way. But continuing with what we've got here, oh, I lost my text box. Law of segregation and law of independent assortments. In the independent assortment, I can spell, guys, I promise. So genetic variation, very important. The second thing that meiosis is going to do for us, it's going to produce haploid gametes, which again are our sperm or egg cell, so that the parent DNA must, I can't spell, must be combined to make the kid. The last thing we want, and this isn't a knock against anybody in specific, but the last thing we want is a second one of you. We don't need somebody who's just like you. We already have you. We're not replacing you. We need people who have new combinations who might be capable of different things. Variety is what actually keeps us alive. Now, I want to talk a little bit about independent assortment real quick. Independent assortments. Independent assortment is homologous chromosomes line up or line up in prophase one. The order they stand in is random. Now, I know that that doesn't really help as a definition. So we're going to go to a picture. We're going to check this out. We're going to specifically go on here and Google prophase one. Actually, we could do metaphase one. It might make it a little bit easier. Images. Looks like I'm going to have to clear my screen. Wow, a bunch of these are not metaphase one. And there's a really easy way I can tell. In metaphase one, the chromosomes should still be next to each other, not in one single file line. We'll use this picture here, I guess. Let me clear my screen because this annotation's in our way. Now, if you look at what we've got here, they lined theirs up sideways, but that's not a big deal. Notice what we have. We have the mom chromosomes on this upper side and the dad chromosomes on this lower side. Now, we said in our definition that it has to do with how they line up. If what I'm trying to say is it's possible that these line up on opposite sides. Most people, your parents have a side of the bed that they sleep on. Oh, yeah, dad's always on that side of the bed, mom's always on that side of the bed. Well, that's going to be individual to you. Somebody else's parents might sleep on opposite sides compared to the side that your parents sleep on. So that's going to be different. I'm actually going to move into another picture here because I see another one. So this one had all the moms on one side and all the dads on the other side, but check out this one. I can't zoom in. This one had mom on the top, then dad on the top, then mom on the top. What I'm saying is when they line up next to each other, who's on the left and who's on the right is technically random. Yeah, I'm still not seeing very many good diagrams here for this. Let's actually go to Google. Maybe I can't Bing things. Still pretty similar, although this one still works. You can see in our diagram here, this one, mom on the top, dad on the top. If these orders were to flip-flop, like if I was to actually switch the locations of these two, if we do that, the breakdown would end up different. If we switch those two, then this top cell that gets produced would end up with 3 copies of mom information. The way it is now, I would get 2 copies of mom information and 1 copy of dad information. But if I flip-flopped one of these other ones, the mom information would be for certain genes because it was that chromosome instead of a different chromosome. Long story short, the order that they line up in is specifically going to tell us the combinations that can get made. The assortment has to do with spreading or putting them into groups. The independent part means just because the first group went dad mom does not mean the second group will go dad mom. It is technically independent of the other group. So that's another way that we can actually get variation within our populations. A human diploid cell has 46 chromosomes. Determine how many chromosomes an egg cell would contain and explain how you know. Okay. So a human diploid cell, here's the cell. It has 46 chromosomes. We said this is diploid. An egg cell and a sperm cell. Now they're talking about the egg, but I'm still going to draw both of these. We have an egg and a sperm. Eggs and cells and sperm cells have 23 chromosomes each. These are considered haploid. Now, the reason this is significant is because we need them to be 23. We need them to be haploid. An egg cell has to have 23. The reason is when we add these two cells together, our zygote, our fertilized egg cell, would then end up with 46 from the 23 from the egg and the 23 from the sperm. If the egg already had 46 chromosomes, then it wouldn't need a sperm cell to fertilize it, and it would just be making copies of a diploid cell. That would be like mitosis. That would be giving me identical copies of the same thing. We do sexual reproduction on purpose, where we use meiosis to get down specifically to haploid egg and sperm cells so we can do this 23 plus 23 equals 46. This zygote is now going to start doing a bunch of mitosis and turn into the baby. So when we say things like body cells are diploid, the reason your body cells are diploid is because the original cell that made you was produced from an egg and a sperm being combined, which added up to that 46, and that's why that's the normal number for your cells. Continuing to our chart down below here, compare and contrast mitosis and meiosis. It is very, very high likelihood that if they ask you questions about this, about mitosis or meiosis, that it will be in reference to the other one. So this chart is definitely something you're going to need to review, definitely something that you would want somebody to quiz you on just to make sure that you know what's going on. But again, all we can do is tell you, hey, make sure you take care of business. This is one you would want to take care of. If you're the person who can take a warning or a hint, this is one to be aware of. So mitosis and meiosis. The first, they ask sexual or asexual reproduction. Sexual involves two parents. Asexual involves one thing copying itself. Mitosis is about taking a body cell and having it produce another copy of that same exact body cell. So this one is going to be asexual. And meiosis, that has two parents because of egg and sperm cell, is going to be sexual reproduction. Number of divisions. Mitosis is going to divide one time. Meiosis divides two times. Haploid or diploid. Mitosis is going to go from a diploid cell, make a perfect copy to end up with a diploid cell. So diploid. On meiosis, it's going to start with a diploid cell and then go through two divisions to make haploid cells. So meiosis is about haploid cells. Number of cells produced. In mitosis, we do this one division we mentioned here, and we go from one cell to two cells being made. In meiosis, we divide twice, so we went from 1 to 2, and then 2 to 4 cells. What type of cells? Well, mitosis is going to be about making somatic cells, which is our body cells. Meiosis is going to be about producing gametes, which are our sperm or egg cells. Mitosis. Mitosis is about making cells for your body. We want them to match. So this is going to be making identical cells. Meiosis is all about genetic variation. So we're making different cells there. That's why we had independent assortment and crossing over and all that. Crossing over, we just mentioned with meiosis. So yes, it definitely happens. Yes, during prophase 1. Does crossing over happen in mitosis? I wouldn't want it to. I want a perfect copy of that original cell again. So does crossing over happen in mitosis? No. Now, when we talk about 2N or 1N, that N is referring to the number of sets of chromosomes that we have. Since mitosis is working with diploid cells, that's both sets of chromosomes, the mom and dad version. So this is going to be 2N. Whereas if you were producing sperm or egg cells, you are only one half of that equation, haploid cells. So this would only be 1N. Definitely take a look at this chart. Very important to the questions on the test. Clearing this out, we scroll down for a little bit of vocab here. Couple things to keep note of. Nothing crazy. We've kind of been talking about it. Meiosis, cell division for making gametes. / reproductive cells. Stem cell. We didn't really talk about this one specifically, but let's make sure we bring this up. A stem cell, if you look it up, would say an undifferentiated cell. This cell has not gotten a job yet. It can turn into any type of cell. These are usually associated with babies. Stem cells are the things. They're the cells before they've become a liver cell or a skin cell. They have the instructions for everything. They can become whatever they need to become. Stem cells are a big area of research because stem cells, if we could use them or activate them, could help us regrow things or grow new organs for you that we don't currently have. Gametes. Again, this is our sperm or egg cells, and they are specifically haploid. One set of chromosomes. Body cells, just reminding you that those are referred to as somatic cells. Diploid, 2 sets of chromosomes from mom / dad. Haploid, 1 set of chromosomes from mom or dad. 2 left, crossing over, and homologous chromosomes. Trade arms or legs or variation. And our last one is tetrad. Tetra means 4, so let's get that on here. Tetrad means a group of 4. In our case, homologous chromosomes are the tetrad. And again, I'll show you that in a picture when I scroll up above in a second. The reason this is significant, 2 copies of 2 chromosomes adds up to make up that 4. If we look at our picture up here in our homologous pair, I end up having 1 copy of a chromosome, a second copy of it, a third for the 1 copy of that, and a 4th for the copy of that. This homologous pair is a group of 4. This also happens during prophase 1. So if they ask you when stuff happens in in meiosis, more than likely the answer is prophase 1. But yeah, the tetrad is that group of 4 chromosomes that line up before division in meiosis 1. You guys, you did awesome. Only got 1 more video left. You can do this. Review your stuff. Don't give up. Don't say it's too hard. Don't decide to only look at certain spots. Review everything. Most of you should have gotten your feedback cards. Look and see which topics you need a little bit of extra work on. Either watch the video again or run back through the review. Hopefully this was helpful. Catch you in the next video. Later, guys.